Hello everyone, my name is Varun Sondhi. I'm currently a resident in internal medicine at the University of Texas Southwestern Medical Center. I recently completed the physician scientist training program also at UT Southwestern and obtained my PhD in the laboratories of Dr. David Mangsdorf and Dr. Stephen Kleber. Today I'd like to talk about our manuscript in molecular endocrinology on work done in collaboration with Dr. Richard Arcus, now at the University of Michigan but previously also at UT Southwestern. Our work focuses on cytochrome B5, a small hemoprotein which is an important cofactor for various P450s. These P450s are involved in a host of different reactions, from steroid and fat metabolism to the catabolism of xenobiotics and drugs. We focused on the role of B5 in regulating the lyase reaction of CYP17A1. CYP17A1 is an important enzyme in steroidogenesis. It functions to catalyze two different reactions, the 17-hydroxylation of progesterone and pregnenolone, and the 17-20 lyase reaction, which cleaves these C21 steroids into the C19 steroids, such as androstenedione and DHEA. Now, B5 only stimulates the lyase reaction, but has almost no impact on the hydroxylase reaction. To further study the effects of B5, in an animal model, we created Leydig cell-specific CYB5 knockouts by crossing flux B5 mice within a Cree driven by SF1. We were surprised to see that these mice were more than normal Mendelian ratios and had no overt phenotype. We found no differences in testes weight, testis histology, sex organ weight, body weight, and most importantly, no differences in fertility. When we measured basal serum steroid levels, we found no differences between the B5 knockouts and the wild-type littermates. However, when we took the testes from these mice and created testicular homogenates, and then in vitro tested for the lyase and hydroxylase reactions, we actually did find that the CYB5 knockout mice were deficient in CYP17A1 lyase. We then added back recombinant B5 and could actually restore the lyase reaction back to the same levels as the wild-type mice. So we did know that there was a defect in the CYP17A1 lyase reaction, so we proceeded to figure out why we weren't seeing any physiology. Now, mice are different from humans in one regard. They have very low basal testosterone levels, and when you expose them to a receptive female, they get a big spike in testosterone. To simulate this effect in a more controlled manner, we injected these mice with HCG and then assayed serum steroid levels 2 hours later. We now found a profound defect in the CYP17A1 lyase reaction. We found an accumulation of substrates upstream of this reaction, especially 17-hydroxyprogesterone, which was 100-fold higher in our knockout mice when compared to the wild-type littermates. Testosterone levels were also lower in the knockout mice, about 25% of the stimulated testosterone levels of the wild-type littermates, but it wasn't as pronounced as the accumulation of 17-hydroxyprogesterone. So we feel that the increased substrate was pushing through the reaction despite the defect in the 17 lyase reaction caused by the lack of B5. So in conclusion, cytochrome B5 is important in the CYP17 lyase reaction, both physiologically and in vitro. However, mice are probably not the best organisms to study the physiology of B5 mutations and knockouts because they virilize and are fertile at much lower levels of testosterone. I thank you for listening, and I hope you enjoy reading our manuscript.